Okay, so 8.4 is all about enzymes and what they do and how they work. So the first thing we need to remember is that a catalyst is a biological agent, at least in biology, is a biological agent that speeds up reactions, but they're not used up in the process. So even though they are speeding up chemical reactions and they're very much involved in the process, they do not get used up, destroyed, degraded, or changed in any way. They can continually do their job. So enzymes then are proteins. Okay, so they belong to the macromolecule class of proteins. But an enzyme is a special kind of protein. It is a catalytic protein, and they do various functions. And a good example of a catalytic protein is sucrase. And all enzymes work in the process of breaking things down. At least those enzymes that are involved in decomposition break things down through hydrolysis. And so hydrolysis, using a water molecule, if we take a look at that on this page, sucrose, you can see right there, is our disaccharide molecule formed from the joining of our two monomers, glucose and fructose. But once you have sucrose, you can break it back apart. And we talked about hydrolysis as the opposite of dehydration synthesis back in chapter, 5, I think. But sucrase then is the enzyme that speeds up the process when water is applied to break apart that bond, this glycosidic linkage between our glucose and our fructose. So sucrase is present to help speed that process up. So we know that there is always an energy requirement. We know that there are exergonic reactions which release energy, and there are endergonic reactions that require energy. But every reaction, whether it's overall exergonic or overall endergonic, all reactions require a certain type of energy to get started, and that energy is what we call activation energy, or the free energy of activation. So whether we are bond breaking or bond forming, whether we're releasing or gaining energy, all reactions require a bit of a push to get started. So we symbolize that with E sub A. Oftentimes, activation energy for a reaction can be in the form of heat from the surroundings. Like even in our lab today, if you were to take starch and let it sit out overnight in a small quantity of it, or let it sit for a day or two, you could come back and test that for the presence of glucose and potentially find glucose in there, because that sucrose would absorb the heat from the surrounding environment, and that would speed up the process, the natural decomposition process of hydrolysis. So activation energy is the amount of energy we need to get a reaction started, and it can be in the form of heat. So you'll see on this image, this is a activation energy, or an energy flow diagram. When we're looking at our reactants, which obviously, we're looking at this diagram is showing us an exergonic reaction, right? Because our reactants have more energy than our products end up with. My delta G is -0, which we know is an indication of an exergonic reaction. And you can see the activation energy that is required to get the reactants to break apart, right? To convert. So notice the amount of activation energy required in this non-enzymatically controlled reaction. If an enzyme is present, that amount of energy, that hump that is our activation energy, would be lessened if an enzyme was present to get our reactants to their transition state. So enzymes catalyze these reactions by lowering that amount of energy. However, if you noticed in that picture, what did not change, or what will not change, is the amount of free energy that the reaction produces or uses. So no matter what enzyme you use, or no matter if you use an enzyme, the amount of free energy does not change. It simply speeds up things that would happen naturally. So here we see that same image, but now we've overlaid a line here that shows us what will happen if enzymes are present. So we have our typical without enzyme line. You can see how high that activation energy hump is in that lighter part of the graph at the top. But when you use an enzyme, our reactants still start at the same point, our products still end up at the same point, so the free energy is the same. The free energy of an enzyme catalyzed versus the free energy of an enzyme uncatalyzed reaction stays the same. You do not change the energy, the free energy. What does change, though, is the activation energy, and you can see that difference in the light part of the graph. Activation energy without and activation energy with is obviously much lower. So enzymes then have, very specific names. They end in ACE, but all enzymes are specific. Okay? They work only on certain substrates, which is why we can get certain names for things like sucrase breaks down sucrose. It doesn't break down maltose or lactose or any other kind of disaccharide. It only breaks down sucrose. So substrate then is the reactant that our enzyme acts on. Okay, so when the enzyme binds to its substrate, you get something called the enzyme substrate complex, and that simply lets us know that we have a reaction, an enzyme catalyzed reaction going on. The active site is the location on the enzyme, and again, remember, enzymes are proteins, so in that complex three-dimensional structure, you have a groove or a hollowed out area, a location on the protein, on the enzyme, where the substrate will bind. And it's only there that our substrate will bind to it, and that active site is specific for the substrate. Not any substrate can bind to that particular active site, only the one that that enzyme works on. When the substrate binds to the enzyme in the active site, the protein or the enzyme, again, this is a very dynamic process, and a protein is kind of a globular, or this enzyme's kind of a globular, think about it like a blob kind of thing. So when the substrate binds to the enzyme, what happens to the enzyme is a phenomenon that we call induced fit. So the enzyme literally kind of squeezes around the substrate. So when the substrate binds to the active site, that will enhance the active site's ability to catalyze the reaction, because literally the enzyme will tighten its grip, if you will, on the substrate, putting stress on the bonds of that substrate and speeds along the process. So here we see our blue enzyme. The active site is that little groove, the substrate is the red molecule there, and when that substrate binds to the enzyme substrate complex is undergoing induced fit. So it squeezes around the substrate, creating stress on it. So that enzymatic reaction is the catalytic reaction of the enzyme and the substrate. Again, the active site is what does the work. It will lower the activation energy, and it does so by doing the following things. So how does that active site lower the activation energy? Well, it will orient substrates correctly, means it will get them in the right position. It will put stress on the bonds of a substrate, because we know unstable bonds react faster. It will provide the right microenvironment. So within that active site, perhaps the pH is ideal, or maybe it's the right polarity or the right charges or whatever it takes to make those substrates break down. And the active site will covalently bond to the substrate, which helps kind of break apart the substrate or strengthen then the bonding between two substrates, if that's the case. So here we have our process. My substrates are the red and the green molecules. They fit into the active site of the enzyme. That active site lowers the activation energy. At step 4, you see our substrates being converted to products. They get spit out of the active site, and the enzyme goes back to its original shape. Enzyme is not affected, not changed. It just constantly keeps pumping out product. There are things that can affect the enzyme's activity, like environmental factors like temperature and pH, because remember, proteins are very complex structures with a lot of bonds holding that structure in place. So if you change the temperature or you change the pH, you can affect the bonding of that protein, which is going to affect its three-dimensional structure. And if you affect the three-dimensional structure, you're going to change the job of the enzyme. And obviously then there are chemicals that will influence the enzyme as well. Again, optimal temperature and optimal pH. All enzymes have that. Different ones have different optimal temperatures and different optimal pHs that are different from ours. This graph is showing us that we see two enzymes, one that is a human enzyme and one that is a enzyme from a heat-tolerant bacteria. Obviously, they have two different optimal temperatures. Ours is around 38 degrees Fahrenheit, or body temperature, and the heat-tolerant bacteria has an optimal temperature of around 78 degrees Celsius, so much hotter. Same thing for optimal pH. Our stomach enzyme, pepsin, has an optimal pH of around 2. So if it gets too low or it gets too high, pepsin doesn't do a good job. Trypsin, which is an enzyme found in our intestine, has an optimal pH of about 8. So again, different enzymes in different parts of our bodies with different optimal pHs. Last little thing to talk about here is that there are things that can help enzymes. Cofactors are one of those. Cofactors are non-protein helpers. So they can be inorganic, like metals in an ionic form. Iron, for example, is a good example of a cofactor. They can also be organic. If it is organic, if it's an organic cofactor, we call it a coenzyme, like coenzyme Q. Maybe you hear about those kind of things. You see them in health food store aisles that you can take supplements for, but coenzymes include many vitamins as well. So cofactors are inorganic helpers. Inorganic inorganic helpers. They're non-protein molecules, metals and such. Or cofactors could be organic, like a coenzyme in vitamins.